So chapter 15 and 16 are both trying to integrate what we're learning about the nervous system. So I really liked this image because it shows us that 15 and 16 are both about neural integration, but it's also showing us how these chapters are kind of being separated. So here in chapter 15, we're going to focus really on two main things. That's going to be how sensory information is getting to the brain. So we're going to talk, and I know we've already said that, right? This is the afferent nervous system, but we're going to talk in more detail about what these receptors are like, what we mean by a pathway, right? How this sensory information is actually getting to the brain. And then we'll dabble a little bit into how the brain is actually making sense of this incoming sensory information. We'll also then kind of shift gears and move towards motor output, and particularly motor output that is part of the somatic nervous system, motor output that is going to skeletal muscle. In chapter 16, we'll continue this idea of motor output, but we'll look more specifically at the autonomic nervous system and how we're affecting the viscera. And we'll also really shift gears and talk just briefly about some of these ideas of higher order thinking. Okay? So in chapter 15, we're looking at sensory information coming in and somatic motor output. Okay, so one of the key things that I really want us to understand is this idea of the difference between sensation and perception. Right? And so this image is giving us an idea of how one of our special senses, which we won't really talk about until chapter 17, but one of our special senses here, olfaction, our sense of smell, how it functions, right? So we have a variety of different citrus fruits, the actual chemical molecules that attach to sensory receptors in the nasal mucosa, right? And we see that, I think what they're trying to show us is this one, right, attaching here, gives us this idea of an orange. But that chemical actually has to bind to the receptor, and an action potential then has to make it to the brain in order for us to perceive this orange, right? So there could be chemicals in that room, right? The sensation, we could have this chemical actually binding to these receptors, but if that doesn't trigger an action potential, or if that action potential doesn't reach the cerebral cortex, right? The cortex is where we have conscious thought. That sensation never actually enters our perception, right? And so I think this is really kind of fascinating and also important for us to understand that we do not fully accurately perceive the world around us, right? Here's another one. This is a dandelion as we see it, and a dandelion as a honeybee. Honeybees see into the ultraviolet, and in fact a lot of flowers, instead of being a solid color, kind of have these more directed indicators of where the floral resources, things like nectar and pollen, would be located. So we perceive a solid yellow flower, even though in the ultraviolet there are several colors involved in this flower. And that sensation, right, those UV rays reflecting off the flower are hitting our eye, but we do not have the receptors in that case to do anything with that information, to send it to the cortex, right? So we do not perceive that, we perceive this. So we'll get into some interesting topics as we kind of dive in a little bit further here. Okay. Now, again, as we're looking at how our body deals with sensory info, we've already talked about this kind of superficially. We're going to dive more in this chapter. Again, understanding this idea that there has to be some kind of stimulus that's arriving, we need a receptor to actually pick that information up. Now, again, we are going to talk about the more general receptors in this chapter, so touch, pressure, vibration, proprioception, those sorts of things. We'll leave the special senses till chapter 17. In that case, we have some quite complicated receptors to pick up on that information. Okay, so we need the stimulus to actually activate or depolarize this sensory receptor. Now, this idea of a sensory pathway, this picture doesn't really do it justice. Hopefully it'll make more sense by the time we get to the end of the chapter. But what we're looking at here, right, is that we need that action potential to be both generated, right? So this stimulus has to be strong enough to cause the graded potential on that receptor to actually trigger an action potential. If you don't get an action potential, and they call this step transduction, in case I forget to talk about that later. Transduction is this conversion of the stimulus into an action potential. Right? And this is one of those places where the sensation may not equal perception. Right? We could have a stimulus, it could actually depolarize our sensory receptor, but unless it's strong enough to trigger an action potential that's going to travel along a pathway, a series of sensory neurons to the brain, right? If we don't get that transduction, we will not perceive the stimulus, right? So there's a few different ways where perception and sensation don't always match up. Okay, so this sensory pathway then is how we're actually carrying the action potential towards the central nervous system. And as we get further into the chapter, we will look at this sensory pathway actually being quite complicated. So what we'll see is that these signals are carried on what's called a labeled line. And each labeled line only carries one modality. So it'll carry pain, or it will carry pressure, or it will carry temperature. And so as we're carrying this information, the nerves that it's coming into the brain on make a huge difference in how we perceive that input, right? The example they give you of this is if you rub your eye hard enough, you'll see maybe shooting sparks. So even though you were applying pressure to the eye, right? Pressure was the modality, but that labeled line only carries optic information, right? If you push hard enough that it's actually triggering an action potential on the optic nerve, you'll see these little shots of light, because when that action potential reaches the brain on that particular pathway, that particular labeled line, it's perceived as light, right? Again, sensation versus perception can differ. Okay, we need to carry that information then to the central nervous system for processing. And this can get quite complicated, right? We will discuss this more, but we basically are going to have to do some processing for the somatic nervous system, right? I like this. Here they're pointing out again perception. Only about 1% of that actual arriving sensation is actually turned into perception. In order for us to perceive something, it has to land on the part of the brain, the cerebral cortex, that's capable of conscious thought, right? And then we can have this voluntary response, a somatic motor response, right? We could also have some information that triggers an involuntary response, right? Things that you don't actually have to think about, things like balance as well. But both of those would be somatic nervous system or motor output. Okay. Just to point out here in this processing, how the brain interprets this incoming information has a couple different pieces, right? So it is going to determine the intensity of the stimulus based on the frequency of the arriving action potentials. right? So harder pressure, right? I can touch my finger lightly, that's going to have action potentials come into the brain. If I really squeeze my finger, that's going to send more frequent action potentials. So the brain is making sense of this based on the frequency of the arriving action potentials. Okay. It figures out the modality, right? Was this pain? Was this touch, vibration? What is this? Based on which labeled line it came into the CNS on. And again, this is how some of those things like referred pain or phantom limb pain can become an issue if the labeled line is being stimulated somehow. Even if you don't have that limb, you still might be getting information to the brain that says, hey, this hurts, right, in a particular area. Speaking of which, location, how the brain determines the location of that stimulus is basically where on the cortex that sensory information lands. Okay. We talked previously about that idea of there being that sensory homunculus, and we definitely get into that more in this chapter. Okay, so this is kind of the bigger overview of where we're heading in this chapter. And now we're actually going to dive a little deeper into each of these topics. So we're going to start here with receptors. And I will say this chapter has a bunch of information, and I am definitely narrowing it down. If you're a brain person, if this is the direction you're heading maybe in your career, feel free to dig deeper, but I am going to try to make this a little bit simpler than even your textbook, right? Which is a simplification of what you would get if you were taking a whole class on neurobiology or something like that. Okay, so sensory receptors. We can categorize these in a few different ways. So the first way I kind of want to separate them is between simple receptors and complex receptors. So the simplest receptor that we have is what's called a free nerve ending, right? And these are just the dendrites, right, the dendritic end of a sensory neuron. And they can still pick up information, right? They respond to things like temperature, pressure, pain, right? All those sorts of things, but there's very little specificity, right? And so in fact, we'll see with these very simple receptors, sometimes, right, if you dripped battery acid on your skin or touched yourself with a hot pan, both of those might be perceived as burning, even though the stimulus is very different, right? So these tend to not be extremely specific. The more complex receptors, the really complex receptors, we are going to leave until chapter 17, but they are extremely specific, right? We have these entire organs that are set up based on both the receptive structures, something like the retina, but also different accessory structures that are going to block other incoming information to make sure we really focus on the stimulus that we're interested in. Another way to categorize our sensory receptors is tonic versus phasic. And so basically what they're showing us here is that tonic receptors are always active. Notice what happens. Even under normal conditions, they're showing you an action potential coming to the brain. Now, when the situation changes, the frequency of that action potential changes, right? But these receptors are basically always on, right? So even when it's not stimulated, we see an action potential going to the brain. Okay? This phasic receptor, right, only sends action potentials when things change, right? So nothing going to the brain, nothing going to the brain. Maybe the temperature here increases, boom, boom, boom. We're letting the brain know that temperature stays high and we stop getting information about it. And we don't get information from phasic receptors again until, say, that temperature, whatever the stimulus is, drops, decreases, right? Now, this difference is really important in what we call adaptation. Adaptation is the decreased sensitivity in the presence of constant stimulus. So think about the adaptation here. When we talk about tonic receptors, we say that they're slow acting or that they have little adaptation. Basically meaning these receptors, even when there is a constant stimulus, we're still going to hear about it. One of the unfortunate things we'll learn here is that our pain receptors, our nociceptors, are tonic receptors, right? And so they show very little adaptation. So even after the injury has occurred, we will continue to get information, right? Until really that healing process is complete, we'll continue to get information about the pain that's going on in that location. Now, phasic receptors are considered more fast-acting or quick adapters. Fast-acting, right? So they respond quite quickly to changes in the situation, right? And are quick to adapt. Make sure you can see this. Getting to the bottom of the board, right? So fast-acting, quick adaptation, basically meaning that these do show adaptation. So, and notice how this works, right? You walk into a warm classroom and you notice it's warm because boom, 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 we're getting information sent to the brain about this change that has occurred in temperature. But once you sit in the room for a while, you stop sending information to the brain, and so you kind of adjust to it, right? Unless it's like extremely hot, you're gonna get used to it. You walk back outside, boom, 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 it's cold, and you're like, oh, I should put my jacket on, right? And so that's what we mean by adaptation. When there's a constant stimulus, the temperature's warm, the temperature's warm, the temperature's warm, we're not gonna notice it, right? Because we're typically talking about these phasic receptors. Okay, we'll get into that some more as well. All right, so we had simple receptors, we had complex receptors, we have tonic and phasic receptors. We can categorize receptors based on their location in the body. So we have exteroceptors that are picking up on the outside world, right? So some of our special senses, but also a lot of our sense of taste, touch, pressure, those sorts of things, right? But dealing with our skin, the outside world. We also have interoceptors, right? And these are based in our viscera. So maybe a stretch receptor in our stomach or a pain receptor somewhere in our brain. So a lot of like visceral organs, right? We're checking in on the insides as well. Now, this particular picture lists proprioception there. Your book keeps it separate, so I'll do the same. We're not truly learning about the outside world, so I understand why they're saying inside, but let's keep proprioception separate. This is about the position of joints and skeletal muscle, so kind of your place in the world, where, right, if I lift my arm, I'm getting information from these proprioceptors to tell the brain what the heck I'm doing, right, and why I'm doing it. Okay, so where they're located can differ. And then, finally, what stimulates them can differ as well. So we'll definitely go into more detail on these, right, but these are our pain receptors, temperature receptors. For mechanical reception, we're talking about like physical distortion. Remember we talked about mechanically gated channels on some of our neurons, and then chemoreceptors, so responding to certain chemical conditions. Typically with that, we don't mean dripping battery acid on our skin. We mean things like oxygen levels or pH levels in the blood. Right, so you can see how there's going to be overlap as well between location and what the excitatory stimulus is and all sorts of these factors. Okay, so let's look at these in a little more detail. So our nociceptors are our pain receptors, right? I think your book tells us, I don't remember if it was Latin or Greek, right? But this noxa equals harm, right? These are our pain receptors. These are really common in places like the skin, joint capsule, the periosteum. If you'll recall, that's that covering around the bone. You have some of these in blood vessels, etc., right? Our nociceptors are not particularly specific. So again, this is where we were talking about that idea that you might burn yourself with a hot pan or a chemical or even do mechanical damage, and you still might feel that sensation, that pain might be this like burning sensation, right? A similar feeling, particularly if it's a kind of a strong stimulus. Nociceptors are a classic example of a tonic receptor. Which again means they don't adapt, which again, unfortunately, means that we're always feeling this pain, right? It's kind of this constant reminder since we have very little peripheral adaptation. And a lot of us have experienced that, right? You may have sprained an ankle and it's still bugging you. You still feel the pain long after that initial damage is complete. When we talk about pain, we actually have different types of pain, and a lot of this has to do with what type of neuron is actually carrying that pain signal, right? So we talk about type A fibers and type C fibers. Okay. One of the big differences here, type A fibers are myelinated. And then, as you might expect, they're able to conduct that action potential more quickly. And so sometimes we call this fast pain. And more than just that it goes to the brain quickly, we're often able to much more pinpoint the area where that pain is emanating from. Type C fibers are unmyelinated, and so these, sometimes the pain perception that we get here, they call slow pain, and a lot of times this is more of a vague area. You might not be able to pinpoint exactly where that pain is coming from. There's other things that go into how specific you can get in describing pain, but one of the big differences is which type of axon is actually carrying the pain message. A couple other things that are kind of unique maybe with nociceptors. So nociceptors are actually going to use different neurotransmitters. So instead of using acetylcholine, we're going to use glutamate and substance P. As our neurotransmitters. I suppose this is nice that we're not using the really common acetylcholine. Hopefully we're not confusing other messages other than pain as that's traveling through our body. It does get kind of interesting. The book goes into, one of the nice things is that you can use things like endorphins. And if you're not familiar, endorphins are often released by things like aerobic exercise, right, going for a run, you release a neurotransmitter called endorphins, and that can actually block the release of substance P, right? That can actually decrease pain sensations from other parts of the body. Obviously, if you sprained your ankle, going for a run is not going to make it feel better, but maybe you might not notice, I don't know, you've had this hangnail that's bugging you, it's just hurting, and you go for a run, and those endorphins can potentially block the release of that substance P. That's also then where we get the use of drugs like morphine, right? Endorphins, just, it's a kind of a contraction for endogenous, endogenous morphine. So morphine that's basically made within your body, where morphine is this drug that we can use to act similarly and block the pain by reducing the release of that particular neurotransmitter. So you can decrease the perception of pain. But notice there's another sensation versus perception. The receptor might still be sending the signal, but if you block the neurotransmitter release somewhere along the line, you might be able to interfere with the message actually making it to the cerebral cortex where you would perceive the pain. Okay. Moving on here. So our next group of receptors are the thermoreceptors, and as you might guess, these do in fact sense temperature. Right? Most of our thermoreceptors are going to be free nerve endings. Right, which we pointed out are not particularly specific, right, in what they're receiving. So you have a lot of these free nerve endings, right? You put some of them up in the dermis, you have them in skeletal muscles, the liver, right, one of your big internal organs actually has some thermoreceptors. Where else was I thinking? So dermis, that'll help you sense the outside temperature. Liver, skeletal muscle. So as you start exercising and things are warming up, and then hypothalamus is who I couldn't come up with. And the hypothalamus is a region of the brain that's actually in charge of a lot of things, but it sits there and actually takes the temperature of your blood, and this is going to be, the hypothalamus really acts as your thermostat in your body for deciding, okay, when should we sweat, when should we shiver, like how do we maintain a constant internal temperature. Temperature receptors, thermoreceptors are kind of our classic phasic receptor. Right, meaning they do show a lot of adaptation. So this idea that you might feel the temperature change when you first walk outside, but again, as long as it's not super extreme, you would actually kind of get used to that. You would stop sending that information onto the brain. Now it does seem we are much more sensitive to cold. So turns out we have 3 to 4 times more cold receptors than we do heat receptors. I don't know if that's true about kids born here in Steamboat. They run around in shorts year round. Maybe they don't have the same cold receptors. The other thing that I'd point out is a lot of this information we don't let into our consciousness in the fact that we'll send this sensory input from thermoreceptors up to the thalamus, right? So we're in the brainstem diencephalon area, lower portions of the brain, but then we don't always send that on to the cerebral cortex, again, where it needs to go if we want it to enter perception. Okay, more sensory receptors. We also have mechanoreceptors. Right? These are the ones where we said the stimulus is actually going to distort the plasma membrane. Remember we had, oh goodness if I can draw this, mechanically gated channel embedded in the membrane. And so if you, say, pushed on this membrane in a way that actually distorted it enough, I so can't draw this, right, distorted it enough, you would actually open the channel, and that's what's going to then lead to, say, our depolarization. There are a variety of receptors that are mechanoreceptors and work based on distorting the plasma membrane, right? So that's the big thing here. The stimuli distorts the plasma membrane. And the easiest one to think about probably is like touch, right? As you're pushing on that receptor, right, or pushing on that cell membrane, it might open this channel. But we also will see that pressure receptors inside of the body, right, baroreceptors and proprioceptors work this same way. So let's look a little more at our tactile receptors. And we have a huge variety of these. Sorry, that's not cleaning very well. So this is a nice figure out of your textbook that shows you a variety of different tactile receptors. Here's that free nerve ending that we've talked about multiple different times, right? A free nerve ending gives you information about things like touch, pressure, pain, and temperature. Now notice, we've already talked about nociceptors, and here we are in tactile receptors. And so what I'm pointing out is this free nerve ending. It is a tactile receptor, right? It tells us things about like touch and pressure, but it also could be considered a nociceptor or a thermoreceptor, right? So these are really the tactile portions of that input. We also get information on the surface of the skin. If you think back to what chapter 5, I think, was the integument, this root hair plexus. So this kind of bundle, these nerves kind of wrapping the root of a hair, so that when the hair is displaced, we can get information from that. We have what are called tactile discs, where we have a tactile or Merkel cell sitting on top of a free nerve ending. Right, so tactile discs or tactile cells, we saw that in that stratum basal, again, of the integument. This gives us much more information maybe about fine touch and texture. Right, so picture maybe rubbing the surface of an orange and feeling the dimples, right? Like this would be like, oh, you're touching something, and this would give us that actual feeling of the surface of the skin of that orange. Anyway, other tactile receptors, you have, where's the tactile corpuscle, formerly called a Meissner's corpuscle. They're really trying to get more towards this getting away from people names, right? So I'll go with tactile corpuscle. This gives us more information about pressure as well as vibration. Okay. Who did they go to next? I have all the old names in my notes. Lamellar corpuscle, previously called a Pacinian corpuscle. This lamellar corpuscle, these last few are a little bit more complicated. Notice they look a lot like this free nerve ending, but we start to have other structures kind of wrapped around them. And that's what's actually changing the way we get this incoming information. Okay. So these are doing a bit more things like deep pressure. If they show you here in the picture, yep, notice those are located deeper down in the dermis, so you wouldn't even stimulate them unless that pressure was a little more robust. And then last but not least, this bulbous corpuscle, formerly called the Ruffini corpuscle. I don't know which is better, the people names or, these aren't particularly descriptive terms here. Anyway, this bulbous corpuscle, again, is giving us, they are located deep in the dermis as well, so some more information maybe about deep pressure. So these two are really similar, but basically what we're looking at is they're actually different in how quickly they accommodate. So lamellar corpuscles are actually, you don't have to know this, lamellar corpuscles are more that phasic adaptation, and these are going to be considered tonic receptors. Yeah, okay. Anyway, in our skin, in our relationship to the outside world, we have a lot of exteroceptors. And so even though these aren't considered special senses, notice that you actually have a wide variety of different sensations that you can pick up on here in the skin. Right? And again, these are all examples of mechanoreceptors. So it's that distortion of the cell membrane that's causing an action potential to be propagated. I thought this was interesting. They're also pointing out this idea of what's called a receptive field. And so here they're using free nerve endings to kind of show you the whole area of the skin that if you touched it, maybe with like a toothpick, right, you would pick up on that location versus if you scooted over a little bit where you would feel that. And one of the things they point out is that in particularly sensitive areas, maybe your hands and your face, you tend to have much smaller receptive fields, so you can tell where something is touching you more precisely, where maybe like up on your shoulder or on your back, those receptive fields might be really big, and so you're like, oh, you're touching my back, but it's going to be a little harder to kind of pinpoint where that sensation is coming from, right? It's just not as important as it is maybe where something is touching your hand. Okay. The other group of mechanoreceptors that we mentioned were the baroreceptors, the pressure receptors. And again, we talked about pressure in the tactile receptors, but that was pressure on the surface. So these baroreceptors, we're talking about interoception here, so sensing the inside world. And you have a variety of really important ones. Right, so we can tell things like when the bladder is full, when the stomach is full. Those we probably, maybe enter our consciousness. Some of these other baroreceptors, ones that are sensing things like blood pressure or maybe how expanded the lungs are, that's not really going to enter your conscious thought, but are super important. The ones that you'll probably see the most if you continue on in AMP2 are these baroreceptors that sense blood pressure. These are going to be some of the most important. And I did want to point out that the two main locations of these baroreceptors are in your carotid artery and in the aortic arch. And so they talk about this carotid sinus and aortic sinus, and this is where there's a cluster of these baroreceptors embedded in the wall of that blood vessel. I point this out because the baroreceptors are located in these sinuses. We'll talk about chemoreceptors located in the same locations that are sensing things like pH or oxygen levels, and those are going to be called the aortic and the carotid bodies. Very annoying, because baroreceptor starts with a B. They're not in the bodies, they're in the sinuses. And I think that is some BS, that the baroreceptors are located in the sinuses. If that helps you remember, go with it. Otherwise, just ignore me. Okay, so these baroreceptors are sensing pressure differences. Again, they're mechanoreceptors. And then our final mechanoreceptor are the proprioceptors. And again, we said that these are embedded in joints, in tendons, and in muscles. And I don't know why your book didn't have a good picture here. Probably it goes into more detail. I'm trying to remember if they went into great detail when we were in the muscular system. But so I'll point out here the major ones, the muscle spindles. Sorry, this picture is not great. Or the muscle spindle fibers, sometimes they're called. These are embedded in the skeletal muscle, and they're telling us something about the length of the muscle, right? This was the one involved in that stretch reflex when you tap the knee. This one here, again, you can't see that, is the Golgi tendon organ. Right? And this receptor is really right in there where the muscle ends and the tendon starts, and it's telling us how much tension is being placed in that tendon. We don't want to exceed our capacity to resist that tension, right? We don't want to tear things. And then also in a lot of our joints, right, in the joint capsule, we have receptors. And those are typically just free nerve endings in the joint capsule. But again, we're getting feedback on where is that joint? Is it extended? Is it flexed? Where is it in space, right? Again, these are all mechanically gated channels, mechanoreceptors, that give us this kind of sensory feedback. Who knew? We had so much different sensory feedback. Another group of receptors, right? So we had tactile receptors, we had thermoreceptors, we had mechanoreceptors, and finally, we went into a lot of detail on some of those. Finally, here we are at chemoreceptors, right? And these are picking up, of course, on different chemical stimuli. The chemoreceptors tend to do a lot of adaptation, and so particularly if we're bringing in external information, things like smell, we adapt to that pretty quickly, right? A lot of the chemoreceptors that we're really concerned about are these internal ones, and again, see, here we go. Chemoreceptors are found in the bodies, right? I don't know, I didn't make this up. Again, these are just embedded in the wall of the carotid artery and the aortic arch so that we get good information about things like CO2, O2, and pH. Okay. This one they're showing you, we're also sensing changes in that case. These are in blood. That one is in cerebral spinal fluid. Okay, so that's the receptors. Holy cow, hit pause, take a break, right? Because we just finished this idea of how does that receptor actually depolarize? And now we're gonna look more into this sensory pathway. Okay. So we're gonna take a look at the sensory pathway and our CNS processing. Now, the sensory pathways, this is definitely a section of the chapter that I am going to narrow down a bit. We're basically going to be looking at different regions of the white matter in the spinal cord. Because remember, the white matter is the myelinated axons that are actually carrying this information. In this case, these are all going to be ascending tracts, right? Bundles of axons heading towards the central nervous system. You have a really nice picture, 15.8, that is about the level of information that I want you to get as we look at these three main pathways. So before we jump into that, let me just point out a couple of these are a little bit easier to make sense of than others, right? Spinothalamic, right? This is basically telling you this tract, right, it's coming from the spine towards the thalamus. That's easy to see that that is sensory, going up towards the brain, right? Or spinocerebellar, from the spine to the cerebellum. That one's easy to see that this is a sensory or afferent pathway. Posterior column doesn't really give you that same information. Okay, so let's look at these 3 sensory pathways. So one of the things that I should point out before we dive in here is when we're dealing with sensory pathways, we end up talking about first order, second order, and third order neurons. And they're being nice here by color coding those for you, right? So here in red, we're seeing our first order neuron. In white, we see the second order neuron traveling up to the thalamus. And then here, carrying this information out to the cortex, we see our third order neuron. And so what we're looking at is this first order neuron. This is the sensory neuron that's actually delivering this information to the central nervous system, in this case, the spinal cord. Right? If you recall, our sensory neurons were those unipolar neurons, the cell bodies off to the side, and there it is in that dorsal root ganglion. And this is really common, right? That the cell body for this first order neuron is in that dorsal root ganglion. Okay? So delivering this to the CNS. Our second order neuron, then you see the synapse. They point this out, but they keep pointing out there's no known reason, but you get all this crossing over or decussation where this second order neuron actually crosses. So we know that sensory information, say from the right hand side of the body, is going to go up to the left side of the brain, and motor output crosses over as well. So weird, I don't get that into it because they keep saying, well, we don't know why it happens, right? But we do get this crossing over, right? And that second order neuron then is going to be taking this information up the spinal cord. It crosses over and typically is going to the thalamus. There's other regions that it could go to, but if we're talking about sensory information, that's going to enter our consciousness, it's going to the thalamus. Right? So this is considered an interneuron. Right? This interneuron is in the spinal cord or the brainstem. Right? And it's going to the brain. I'll go ahead and put thalamus in parentheses because it's not always the thalamus, but that's the way we're primarily going to think about it. This is the one that does decussation, that crossing over. Whoop. Okay, and then our third order neuron, right, is the one up here in the brain, in this case going from the thalamus, but this is the one actually going out to, again, if it's going to enter consciousness, the cortex. So this third order neuron it begins at the thalamus. I suppose it technically is also an interneuron, right? It's going to find somewhere to drop off that information, but it's not a motor neuron carrying that out yet. Okay, so first, second, and third order neurons are what we're going to see in sensory pathways, right, carrying this information up. Now the one that I'm drawing on here, this is a picture showing you the spinothalamic pathway. I don't know if I have a... Okay, so that spinothalamic pathway, right, again, carrying sensory information up, it's obviously going to the thalamus, so this is going to be the information that will eventually head out to the cortex, right? Sorry. There we go, spinothalamic pathway, right? First order neuron into the spinal cord. This is the one I was drawing on. second order neuron carrying that information to the thalamus, right? The thalamus is that big sensory filter for what makes it via a third order neuron out to the cortex. And again, here's that sensory homunculus again, that the brain interprets where the information is from based on where it lands on the cortex. So you don't want the wiring to get crossed here. Okay. Our posterior column pathway, right? Again, we see this first order neuron coming in. In this case, it's actually happening, notice here we are in the medulla, so we know we're in the brainstem, right? But our second order neuron, there's our crossing over. We go to the thalamus and then on. Let me give you some notes here, sorry. Back up this train. Spinothalamic pathway. And again, you should know these three pathways are carrying sensory information. I am not going to ask you for these excuse me, individual little tracts. Right. But you should know that the spinothalamic pathway is sensory. Right. This information is going to the thalamus to be filtered to the cerebral cortex. Right. And it tells us to the thalamus, where it's filtered to the cerebral cortex, right? This ends up carrying a lot of information about things like vague touch, right? They're telling you here, things like vague touch, pressure, pain, temperature. Okay, and again, recall this idea that where it lands on the cortex tells us where it came from in the body, which labeled line it comes in on tells us which of these is being felt at your big toe, and the frequency of the action potential tells us how intense that particular stimulus was. Okay, so that's the spinothalamic tract. The posterior column pathway. This is also carrying information that is going to make it to our consciousness. Right? It's carrying sensory information. We see it going out to the cortex, so we know that is going to enter our conscious thought. This carries more information about fine touch, vibrations, and proprioception. Right? So they're actually pretty similar, they're just carrying slightly different modalities of information to the brain. And finally, spinocerebellar. Now this one again is telling you it's carrying things up to the cerebellum, but if you recall, the cerebellum is not part of our conscious thought. So the spinocerebellar is carrying sensory information to the cerebellum. Right? Tells us that in the name. This is going to be information coming really from a lot of our proprioceptors. Right? So again, joints, muscles, tendons, not entering our conscious thought, just going to the cerebellum to inform some of our movements. Okay. So again, a lot of information there in chapter 15. that is just the sensory portion of what's going on here. There is a lot of detail, hopefully not completely overwhelming. I will point out, right, all of those pathways that we just talked about are really carrying somatic information. There are visceral sensory pathways as well. Right, carrying information from those interoceptors. Right, monitoring all of those internal tissues, internal organs. Again, these can be nociceptors, thermoreceptors, baroreceptors, chemoreceptors, right, anything like that. They are less abundant, right, than we find, say, on the surface of our skin, our temperature receptors and those sorts of things. And this information is typically going to the medulla, medulla oblongata, right, where we make lots of different connections unconsciously. The medulla takes care of a lot of adjustments in the cardiovascular system, the respiratory system, etc.